Good afternoon, everybody. A little bit of background about my research interests. Initially, my work is mainly focused on human genetic variation analysis. So we all know the holy grail there is to identify disease genes, genes that have, causing disease. So then the question is, what's going to be next? What we can do computationally, once you identify some disease genes, how you're going to utilize this information. So this leads to a different direction in my research. Basically, I summarize as an integration of different data sources, in particular with drug chemical information and disease information and gene expression. A very natural direction is actually how can we computationally integrate those different types of data for drug repositioning. So the model we were using in this research, we call it a heterogeneous network. Essentially, it's a graphic model. We have different layers for different biological entities and different types of links. And then we formulate the drug repositioning problem basically as a missing edge prediction problem. So that's mainly what I'm going to talk about today. Okay. So this is the outline of my talk. So I will give a brief overview of the problem and the need and how people are studying this drug repositioning problem using a network-based approach. And then I will talk about the details of our model and what type of data set we used for drug repositioning using this model, and what the results we have obtained. And finally, I'm going to conclude the work and also very briefly discuss some future work that may incorporate the links data set generated by the links project. So we start with a quote from Sir James Black. He said the most fruitful basis for the discovery of a new drug is to start with the old drug. So this is in general true for anything. You want to do something new, you start with your own thing. But this is particularly true in the drug development area, because we all know the cost of developing new drugs is keep climbing. It's in the order of billions, and in terms of time, more than 10 years normally in order to develop new drug. And the throughput is not proportional to the investment also. There's I'm going to show you a figure first. This is actually from a paper about a decade ago. We see that the increase of research expenditures from bigger pharmaceutical companies. However, the number of approved drugs is steadily. Does not increase at all. Also, the investigation of new drugs, these are not approved, but the potential drugs basically is also does not increase that much. This is in contrast, especially with the knowledge and the data accumulated from the post-genomic area. We know so much basically, but still that data seems does not transfer to new drug development. There are other benefits in addition to with the hope of lowering the cost. If we can find new indications of existing drugs, we have a lot of information already known. So for example, their safety, their tolerance, their efficacy. So basically, you can speed up the cycle of development tremendously. On another note, there are a lot of rare diseases. Pharmaceutical companies do not have much interest to develop new drugs for those diseases because the market size is limited. However, those diseases are also in need of treatment. So we can find some new use of existing drugs that's also going to greatly improve the treatment of those diseases. We all know there are a couple of, there are many actually successful stories. This is a very famous one. And there are also at least a couple of them obtained from different sources. So this is from Food and Drug Administration. Some drugs that can treat both common diseases and some rare diseases. And also this is from another source, the drugs that has been successfully repositioned to treat different diseases. Okay, one thing that for drug repositioning is many of those examples are discovered accidentally. What we want to have is actually a systematic discovery of new indications of existing drugs. In order to do this, we have already accumulated a lot of information in terms of genomics, transcriptomics, proteomics, as well as our understanding about pathways, drug compound and target interactions, their biological activities. So the problem is how can we computationally integrate all this information for better discovery of new indication of drugs. And one common theme of integration of those diverse data sets is through network integration. So there has been a lot of research recently in past several years trying to either study the novel targets or for drug repositioning or for drug side effect, study of side effects by integrating this diverse source of data set. Also, the funding agency like NIH also spent a lot of effort and fund to collect more data about drug properties, drug compound, and target relationship, the activity assays, just like the LINKS project. On the other hand, the larger drug companies, they also very interested in pursue these kind of activities, as evidenced by some of those big companies. They have their own new indication discovery units. Okay, so what are the assumptions for drug repositioning from our study? The first one is very simple. It's guilt by association. Not only us, a lot of previous studies also utilize this assumption. So basically in this setting, we assume similar drugs tend to be associated with similar diseases. This does not mean that drugs from one disease cannot be utilized for a very distinct disease. This is just very natural in terms of there are underlying relationship between among diseases and among drug structures. So this is one of the basic assumption we're going to utilize. Then when you have this in mind, a very naive idea about drug repositioning is to collect information about drugs and to collect information about the disease, and then try to construct a structure called a bipartite graph, and then try to find new links between drugs and disease. That's the first step, we would say, to do this. In our study, we thought it's probably beneficial to directly incorporate the target information into this disease and drug relationship. We hope that by integrating target information, it will improve drug reposition. So that's our second assumption of this study. Then how to incorporate the target information? So here is the idea. So this is the main idea of our research, our approach for this research. So essentially, we're going to construct a 3-layer heterogeneous network model for drugs, for disease, drugs, and target. Within each layer, we have the similarities among same types of entities. For example, we can have disease similarities and drug similarities, as well as target similarities. For different layers, we have links between disease and drugs. This information can be obtained, from drug bank, for example. We also have information between drugs and target. So that's the link between these 2 layers. So our goal is, again, we view this drug repositioning problem as a missing edge prediction problem on this model. So therefore, our goal is actually try to find new links between drugs and disease. Okay. While we are doing this, we may also be able to find new links between drugs and targets. Okay. So that's our basically final output of this 3-layer model. Okay. Then certainly, there are some computational approach needed to how you define the strengths between target, between a disease and a drug. So that's something we have developed. It's basically an iterative algorithm. We're going to discuss that a little bit later. Okay. So this is a summary of that graph. Have 3 layers, each for each entity, and then within each layer, nodes are connected based on similarities. I'm going to talk about how we define similarities among the same type of nodes. And between layers, we have information about the drug target and the drug disease. This is our formulation as a missing edge prediction. So our idea to find the links between drug and disease is through is basically look at the overall topology of the graph. We want to incorporate not only the local topology information, but the overall topology of the whole graph. So the strategy of updating the network is basically iteratively through this formula. This formula is basically the matrix representation of the connectivities of those networks. Essentially, it has a flavor of random walk in networks, but it's a little bit different because we have different types of layers. I'm going to briefly talk about the meaning of this formula, but not in great details. So basically, when we look at the strength of association between a pair of disease and a drug, we define them based on the previous iteration. But we updated them through these formulas. I'm going to talk about what does this mean. At the same time, we gave some weight of the original data, original disease and drug interaction network. We denote as W0. Okay, 1 minus alpha is the weight of the original graph, and alpha is the weight of the previous iteration with some updating matrix multiplication. So these multiplications essentially represent we first through disease-drug interaction and drug-target interaction establish interaction or relationship among diseases and targets. So these multiplications essentially represent the strength between diseases and target. And then we use that information again to update the strength between diseases and drugs. That's essentially what this theory of multiplication did. So while we are doing this, we also need to update the drug and target the strength of their links. So this is actually have a very similar formula compared to drug and disease. Basically, it's very much like a random walk on the graph. We were able to prove that under some conditions, these iterations will converge. So we will be able to find a stable relationship among those disease and drugs and drug and target. That's pretty much about the model and the computational approach. So what data we used? For drugs, we used basically from drug banks, and we use mouse format. For similarity, we use a very similar similarity score, which is also very commonly used. Smart format first to convert to binary fingerprints. For diseases, we use the omics database. Omics database consists many many terms for diseases, and we use previous defined similarities among diseases from previous work called mini-minor. This is a phenotype-based similarity score. It's essentially based on the number of occurrence of matched terms in each disease description. For each disease in omics database, there's a description, and then we look at this approach to look at the number of occurrences of common matched terms from these descriptions. And we find to define the target, we not only including existing known target, we also actually include some predicted targets, or people call it druggable genome. The genes encode proteins that potentially can be targeted. So we include from this database. Okay. The similarity of drug targets is essentially based on the alignment of their protein sequence. So how the initial network looks like? We have included about 1,400 drugs and 5,000 diseases and about 4,000 targets. We're going to take a look at the similarity distribution among the same type of nodes, because we do not want to have a complete graph. We have to somehow select cutoff value to construct a graph. The connections between layers, we have about 1,400 connections between drug and diseases, and about 2,100 drug target connections. So intuitively, this these connections, it's a loose, it's a very sparse because you have so many nodes, but you only have so many edges among those layers. This is actually the histogram of similarities of the disease and drug and targets. We see for all of them, they have a big chunk of distributions that are very small, similarity are very small. So this is a disease, this is a drug, and this target. Okay. So then a problem is to construct a network, what similarity threshold we're going to use. What we did is actually we did a grid search to find some threshold. Okay. When we take a look at the initial links, we found that the known information about drug and disease and drug and target is kind of biased. The total number is small, and also some drugs, some disease might have a lot of drugs. This is a distribution, the number of disease has 0 drugs, 1 drugs, and so on and so forth. Some disease has quite a lot of drugs. And similarly, for drugs, the same thing. Some drugs mostly only treat one disease, but some drugs treat a lot of disease. Okay. The similar distribution observed for target and drug target links. So a lot of drugs only have one target or 0 targets. We don't know their target. But some of drugs have a lot of targets. And also, for some drugs, most of them only have one target, but some drugs have a lot of targets. Okay. So the information is quite biased. Before we do any analysis, we first want to somehow validate the guild by association principle to see whether for this set, does this assumption hold or not? Otherwise, if it does not hold, there's no point doing further analysis. You will not be able to successful. So in order to do this, we basically look at a very simple similarities scores among the same diseases, and we look at the similarity score of drugs among different diseases. And we do observe they are different, and we use Wilkinson rank sum test to test their distribution, and they are different. We also look at, for the same drug, how about the disease similarity, and also for the same target, their drug similarity, and for the same drug, their target similarity compared to, different drugs, different targets, their similarity. So all these similarities, they are significantly different. So if something from the same category, you compare that with similarities that are from different categories, then the similarity of the same drugs or same targets always higher than them from different drugs or different targets. Okay, so this gives us some confidence. Probably for this data set, we can actually utilize this guild by association principle. For our experiments, we first need to select some parameters. Basically, there are 2 parameters. One is the alpha in the iterative updating algorithm. That is how much weight actually we can put on the initial network. The second parameter is essentially for each layer, what is the similarity threshold? Is the cutoff value to connect different nodes together. If the similarity is smaller than the cutoff, there are no edges between them. So we first did some robustness analysis with respect to these two types of parameters. And then for the main experiment, we mainly used the leave one out cross-validation for existing drug disease links. So for each link, it's basically we just mask that link when we do the analysis, and we want to see whether we can recover that link. Another way to say it, for each disease, we rank all the drugs after running the program, and then we want to see the drugs that had the association with the disease or treated the disease, how it ranks in all the other diseases. For this leave one out cross-validation, we can calculate the true positive, false positive, and the ROC curve, as well as their direct ranks. Okay. There's special type of special category of data here, which is different because for all those diseases with a single known drug, if you remove that link, then essentially that disease has no drug. So the algorithm essentially is doing novel prediction to see whether you can find some drug, treat a disease which do not have existing drug for it. This is important because the links between drug and disease is limited compared to the number of diseases we have seen. So we separate this category basically to see if there are no drugs for disease, whether we can predict novel drugs. So this is also we take advantage of the overall topology of the network. If you only look at disease with similar disease that have drugs, this may have some limitations in order to identify those new interactions. Finally, we also did predictions by including all the data sets, and we provided some case analysis for several diseases. As a byproduct, our framework will also provide the drug target, new drug target relationship. So there's a potential of identifying new targets for those drugs. We're also going to take a look at the results. So because this drug target relationship is calculated simultaneously with the drug disease relationship. So in terms of robustness, we did a grid search for two parameters. One is the decay parameter, alpha, in the iterative algorithm. Another is the similarity threshold for all three layers. The decay factor we tested from 0.01, 0.1 and 2.9. And for the similarity threshold, we use 0.1 to 0.5. So the observations are like for fixed decay factors, I think that a similarity score of 0.4 gives the best results. For at the end, because we did this robustness actually after we done our main research. Main research, we actually main experiments. We actually pick a fixed similarity of 0.3, which is actually the same as some previous work. Also, this based on the histogram of this similarity. We actually picked 0.3. Then, when we picked the similarity of 0.3, we found that the model is very robust with respect to the decay factors. The decay factor, the performance is almost does not change much when the decay factor changes from 0.1 to 0.7. Although smaller values, smaller decay factors, which means we have more weight on the original that have slightly better performance. Okay, we choose parameters based on the robustness analysis, and then we compare our approach with a baseline approach, which only consists of 2 layers without the targeting information. The 2-layer model, we also use an iterative updating algorithm, which is very similar to a 3-layer. So we call that a baseline approach. We also found 3 recent approaches for drug repositioning, and we compare our performance with those approaches too. So we call the 2-layer model HGBI, and the 3-layer model it's TLHGBI. And the 3 other approach, one is called the GBA. This is actually a very simple application of guilt by association. It basically says that drugs for one disease, it can be used for another disease if the 2 diseases share some common drugs. So this is a direct application of guilt by association. There are other 2 models. One is called BLM, another called MBI. Both models actually use bipartite graph. The difference of these 2 models, one is use SVM to classify whether a drug and the target, a drug and disease can have a new link or not. The other one is actually use the 2-step diffusion. The difference between this model and our model, we actually use the iterative updating until it's converged. But this one only updating basically twice. Okay. So this is the main results and the comparison of this method. On the left-hand side is the basically RC curve for different approaches. GBA is the algorithm directly utilizing a guilt by association cannot provide a curve. It only provides 1 data point. It's over here. Okay. Then what we can observe over here is actually this MBI, which only have updated the graph twice, has the worst performance actually among all of this. And the 3-layer model has the best performance. The 2-layer model and the SVM model actually have a very similar performance. So what we can observe basically by incorporating targeting information, the 3-layer model is much better than the 2-layer model in general. And the 2-layer model compares this one with the bipartite graph. The iterative updating strategy is much better than the 2-step updating strategy. Okay, so that's our basic message from this graph. On the right-hand side is actually look at the number of retrieved disease-drug relationship when we using different cutoff values. We want to show this in particularly for very small threshold like 1%. If we use 1%, the rank, top 1%, then how many true association, drug disease association can be recovered. We see that both the 3-layer and the 2-layer have significant number of true drug disease relation recovered compared to the other 2 approaches. So this is very is desirable because if you set a very top ranking percentage, then you have a smaller number of false positives essentially. Then when you do experiments to test your prediction based on this result, our approach actually gives more true positives. We also mentioned that for diseases with only a single drug, when you remove the drug, essentially you try to identify novel associations between drugs and disease. we have 79 such diseases. We did analysis on this subset of data. Unfortunately, the GBA and BLM, these two approaches cannot detect drugs, even disease do not have drug itself. Therefore, the information is not included here. They cannot predict no drugs. What we observed here, again, both the 3-layer model and the 2-layer model is much better than the direct application of guild by association. The 3-layer model, it's not a significant different from 2-layer model in this case. Okay. So in this subset, essentially the drug, the target information actually does not provide much improvement. After data delivery the cross-validation, we put all the data together and we run the algorithm on all the data set, and we want to see whether we can identify anything that is interesting. Here we pick 5 diseases to illustrate the result, partial result of our prediction. These 5 diseases include Huntington disease and 2 related cancer, one is non-small cell lung cancer, small cell lung cancer. The other 2 are, one of them are alcohol dependence, and the last one is substance abuse. What we observed from these 5 cases, this table actually represents what are the diseases and what are the known drugs for this disease and what are the top 10 predictions. Okay. By only looking at this table, we can see nothing about our prediction, whether it is meaningful or it is not meaningful. Therefore, we go back to take a look at the subgraphs associated with these diseases and drugs. This subgraph does not include targets layer. So we only have these 5 diseases and the drugs. The original interaction between disease and drugs are the blue solid line. The new predictions are the dashed red line. We only include the top 3 predictions for each disease. Okay. And the green line actually are the similarities among diseases and the similarities among drugs. Okay. So what do we observe from this data? First, these 2 lung cancer diseases, we found that they do share some common predictions. It's over here, these 2, because the reason is because they have some similarities, and they also, even though these 2 lung cancers is quite different, their description still have similarities compared to cancer with other diseases. Okay. Then we found that they do share some common predictions. This substance abuse does not initially have any drugs associated. We predict some of them, and the prediction we believe is mostly because this disease is associated with alcohol dependence. And alcohol dependence, there are some drugs associated with them. Therefore, this substance abuse is actually borrowed information from this alcohol dependence and some of the drugs from these 2. For other diseases like Huntington disease and also this 2 cancer, we did some literature research. We found that the top predictions actually supported by recent literatures. For example, some of those drugs actually are in clinical trials. The information was originally not included in our input data. Okay, finally, we want to take a look at, as a byproduct, we can predict drug and target connectivities. We want to see whether, three-layer model, how it compare with the two-layer model. We separate data again into diseases with multiple drugs and diseases with a single drug. And we see in the first case, actually, these two methods, the three-layer and the two-layer, actually does not have much difference. So this means that disease information may not provide much value to predict novel targets for drugs. There's a little bit difference for the novel prediction using three-layer model. Get a slightly better results from the novel prediction. So this is a byproduct of the drug repositioning. So what do we have learned from this exercise? Something we can conclude basically by adding target information, it clearly helps the drug repositioning prediction. Basically the three-layer model is better than the two-layer model. And also the iterative updating algorithm is better than the two-step diffusion updating approach. Also, the heterogeneous network model actually provided a general framework for integrating different data types. So we were thinking, when we take a look at the links data set, because different assays were generated from different centers, there are a lot of missing. Basically, the compounds from different centers are quite different for different assays. Then these can be viewed as missing. So we were thinking maybe we can actually using this heterogeneous model to predict the missing links between, for example, compound and their targets. Yeah, I already said this. Oh, okay. Actually, so these are basically the conclusions from this study. Is there any limitations about this framework? Certainly. For example, similarity measures, we use very simple similarity measures, which can be improved with more advanced similarity measures to capture the essential structure, for example, the drug, the target structure, protein structure, instead of the sequence level. Another limitation is this model is good. However, what if we have more data types, like a drug side effect or some bioactivity assays? How are you gonna extend this model further to incorporate more different types of data? If you keep adding layers, that's gonna make this model quite complicated and unrealistic. So, so we have to think about how to add in more type of data. Okay. Another limitation is, even though in the paper and in the talk, I try to interpret some of the case studies, the results, but in reality, it's really very hard to interpret all the results because this is an iterative model by considering all the possible connections in the network. Then at the end, when you have the result, you try to figure out what actually contributes to the new link between drug and disease, it's not that straightforward. It's the overall structure or topology of the network. It's not some kind of direct interpretable links in the network. A possible direction to for these limitations is actually try to use semantic networks. So by utilizing semantic networks, you can actually incorporate more different types of data, and also it's if you limit the path, it's not through the iterative updating. You limit the length of path, you might be able to interpret the new links based on the existing links. Okay, there are some people actually utilizing semantic network for target prediction and target repositioning. So when I take a look at the links data set, I think the most relevant data set are those 4 types of data set, including basically cell viability assay, cell growth inhibition assay, genome scan assay, and also the transcription assay. So a direct usage of the links data is actually we can use that to validate our predictions. So for example, when we find some new links between drug and diseases, if it happens that the compound being acid for particular cell line that is associated with the disease, then we can look at how this compound actually there are inhibition activities, for example. And also, if for target prediction, then we may be able to see whether in the genome scan how the compound and target whether the binding affinity. Okay. So one thing we have to consider is again, those assays, the number of compounds, the number of cell lines, and the number of targets may be different from our data. Then another way to say it, there are a lot of missing. Then how can you somehow to tackle those missing and still have some meaningful results about this validation? That is something we need to investigate. There are some other directions when we utilize the network-based models. 2 of them are actually, one is polypharmacology. It's basically, can we take a look at the compounds, whether they actually try to interact with multiple targets. Another similar but different line of research is actually the combination therapy. From the network, can we actually find multiple drugs that can be used to treat one disease in combination? I think based on the data, based on the model, these are doable, and we're going to explore them further to see how exactly computationally we can define the problem and identify the associations. One way to do this, for example, for the combination therapy, there are some existing databases actually store drug combinations to treat diseases. Then we might use that data to learn new combinations based on the public data as well as the links data set together. Okay, finally, I want to thank our students, Wang and Yang, and our collaborators, John. And this research is supported by NSF and NIH grant. So I'll stop here, and any questions?
This is Mario speaking from Cincinnati. I was wondering, maybe you gave these numbers, but I missed them. In terms of actual number of false predictions that you have, say, if on FBR at false positive rate of 0.1, what would that be? I mean, I didn't get the exact relationship between the numbers of true positives and false positives. Right here.
Oh, okay. So it's basically we give a rank. So for each disease, we rank all the drugs based on the association score with that disease. Then we have a ranked list. Then we set a threshold, let's say only using the top 1 drug to see whether this is a drug we try to recover or it is not. If it is a drug that we try to recover, then it's a true positive. If it is not, it's a false positive. Then, varying this threshold, you can say top 5 or top 10 or top 1%. Then you see how many cases actually you can recover the drugs, and also when you reduce the ranking threshold, you're obviously including false positives.
Right, but presumably there are many more false positives than true positives, right? So
yes.
So one,
yeah.
how many would you have false positives, say for 1%?
Oh, so let me see. So for 1%, we have let me see how many links we have. We have about 1,000 links initially. We have how many drugs? Yeah, we have about 1,400 drugs. So then for each disease, 1% is about 14 drugs. We look at 1% of them, then to see how many of them actually the true drugs for that disease.
So that's pretty excellent then. You get 200 true positives for only
Yes.
Positive.
Yes, thanks.
Right, but okay, all right, hold back. Now, this is your true positive totals, right? These are the ones you start with. These are not false positives. False positives are missing links.
You're right. These are true positives among. So basically, these are total number of drugs, and these are true positives. But for each disease, 1% we look at about 14 drugs.
I don't understand that. So your false positives are links that don't exist, right? Because there is no connection between a drug and a disease, right? And true positives are 1,500 known links. Am I right? Or 1,400 known links.
We did this for each disease, and then we sum them up. So for each disease, it only has several true positives.
Right, but it has many. Your false positives are, number of possible connections between disease and drugs, right? So it's like,
yes.
I don't know what this number comes to, but it's huge, right? If you have 1,500 nodes of one kind and 1,500 of the other. So 1% of this number is of interest because that tells you, how many false predictions you made, right?
Yes. So when you see when you take a look at this table, for the prediction like this one, we pick top 10 predictions. Certainly, a lot of them are going to be false positives by nature because the connections is only these two, right? But you look at the rank of these two among these predictions, they are still quite in top. But I agree with you. Many of them, if we only treat the existing knowledge as truth, ground truth, many of them are false positives. But potentially, they may be able to actually, treat the disease.
All right. So,
Does that make sense?
yeah, yeah. So you do get many more. I mean, because in your cross validations, right, you drop the true non-links and everything else is
Right. Right.
false. So the numbers of actual false predictions are still
Yeah, still a
dominating.
lot. Yes, the most of them we treat as the false positive. Yeah.
Okay. And so as you increase, can you show your graph again, the 2-layer versus 3-layer one with false positives? Yeah, that one. So if the difference actually in the 3-layer comes slightly at 5%, right? That's when you see at 1% you don't see any difference really.
Yeah, in terms of yeah, this on the right-hand side, they are a little bit different, but not that much. So this means that including, I think this probably tells us the iterative algorithm actually is much more powerful than the two-step updating.
Right, but there is no additional information gained from including targets, right?
Right, you're right.
Yeah.
At this very top threshold, the difference is it's small.
Right, and then at 5%, I mean, is that specificity you would really use? Because that strikes me as then having a lot of false positives, right?
Yes, yes.
5% of all possible.
Yeah.
Okay, I'm just trying to figure out if I'm interpreting.
Sure.
Well, I have, I don't know, I have one more question if you're willing to stand by me. So, could you summarize very briefly what the intuition of the random walk is underlying your iterative algorithm?
Oh, okay. So it's similar to random walk, but here essentially what we did is okay, maybe it's easier for using this graph. So this is our initial input. Okay. So the intuition is like this. Initially, when you try to predict drugs for this disease, you look at its neighbors, which are similar to this disease, and then you look at their connections over here. Then you predict probably this one also may be able to associate with this one because they saw it together. Even in addition to this one, maybe this one this one can also be associated with this one because this one is close to this one, which this one treats this one is also close to this one. Okay. So that's a direct application of good by association. We say this one might treat this disease. So our thinking is actually, okay, we should include the targets. And how to include target? We first develop the relationship between target and disease. For example, the relationship between this target and this disease is defined based on the intermediate drug information. So basically, the strength of the disease and target, it depends on how many drugs they share, and for those one they don't share, how similar. So let's see which one. This one. Let's say this one share a drug with this disease by direct link. But how about this one? It has a target and drug information, but it has no direct link to this one. Then you look at the drug-drug similarity, and then through this you link this drug with this disease, and then you link the target with the disease too. So therefore, we can first establish the relationship between target and disease through their connected drugs. That can be represented by a matrix multiplication. Once we have that, next time when we updating disease and drug relationship, we actually utilize that intermediate relationship between target and disease. So that's basically what happened over here. This is the we look at the detail. This is the disease-drug relationship. This is the drug-drug relationship. This is the drug-target relationship. Then these three together gives you the disease. The D is for disease, T is for target, disease-target relationship. Then you have this new disease-target relationship. To obtain new disease-drug relationship, then we use disease-target relationship, and then consider target-to-target similarity, and this is transposal of drug-target, and then it becomes target and drug relationship. Then this becomes drug and a disease and a drug relationship again. So that's the meaning of this series of multiplications.
Somebody's asking a question through a chat, Dushitsa. Do you see that question?
The disease similarity, yes. Okay, here. So the disease similarity is from previous work. It's basically the disease definition. We obtain the disease from the omics database. Then for each disease in the omics database, there's a description about the disease. In that description, there are mesh terms. Then you are to define the similarity between two diseases. We basically look at the frequency of co-occurrence of mesh terms among two diseases. So that's how the similarity is defined.
So thank you very much for a great presentation, Jing, and thank you for answering our questions.